the title of my talk today is How Does Activity in D1 and D2 receptor expressing neurons in the dorsomedial striatum influence choice in a non-lateralized task? Now, to unpack what that means, when I talk about a non-lateralized task, I'm thinking more about the type of decision that's being made by this mouse in the wild on the right, who's foraging for food. So guided by olfactory cues, it's exploring its environment and deciding whether to respond to a target or to reject a target and continue exploring and searching for food. I don't have to explain what's happening on the left that much because we've already seen these type of two alternative force choice tasks described in detail earlier in this session. But we can appreciate that there are pretty stark differences in the type of decisions that are being made between these two contexts. So the mouse on the left has to choose between two competing options and signals its choice by making a right or leftward movement, whereas the mouse on the right is mainly deciding whether to respond or reject. So I'm going back to the striatum. We've been talking about the dopamine system. So since the advent of optogenetic technology, especially, experiments that have been done in the dorsal striatum have really stacked up these results that support the idea of dichotomies between the direct and indirect pathway neurons. And we're all probably pretty comfortable and familiar with these dichotomies. So popular ones are go versus no-go. Work from the Wilbrecht lab has shown that activation of D1 neurons unilaterally can promote contralateral bias, whereas activation unilaterally of the D2 indirect pathway neurons can promote ipsilateral bias. It's also been shown that direct activation of D1 neurons in the DMS is rewarding, whereas activation of the D2 neurons in the DMS is aversive. And going along with Sophia's data that was shown in a paper from Rui Costa's lab, now that we can monitor simultaneously the activity of these two pathways, we know that they're coactive before movements. So to update this kind of antagonistic model, we can think instead that the direct pathway might be important for selecting target actions, whereas the indirect pathway might be important for suppressing competing actions. So given all these dichotomies, I was interested in whether we could interrogate them in this more non-lateralized exploratory decision-making context. So we adapted this foraging task to be used in the lab, and it's a very simple behavior. What we do is we train mice to associate one of 4 presented odors with a Cheerio reward that's buried in wood shavings that are scented. And mice start a trial in the center cylinder, and then they're free to explore the arena that has 4 differently scented pots. And they signal their choice by digging into the pot, as you could see that mouse just did. And we train them to a criterion of 8 out of 10 correct consecutive choices. And I should emphasize that this task is not spatial. The locations of the pots are shuffled each trial, so it's really a cue-guided decision-making task. And what we really like about it is that it's rapidly learned, likely because it taps into ethologically relevant behavior. And it's also robust. It produces robust learning after a single training session. So to support this robust learning, I can just show we train mice in the odor reward rule, and then 24 hours later, we can test them for their recall of this rule. And so when we do this in control mice, we see that the vast majority of mice can complete this test phase in the minimum of 10 trials required. So we've run this task in many different mice, and we always use the same odors, odors 1 through 4. And we've observed that mice have innate preferences towards odors, which also makes sense if they're relevant for them in their natural foraging behavior. So those initial odor preferences are reflected in the probability that each of the odors is selected in the very beginning of training. So what we think is happening during training is mice sample the different odors and they receive feedback, either reward in the case of odor 1 or no reward in all the other odors, and that through learning they update these learned values for the different odors. And we think that the learned value weights might be stored in the weights of cortical striatal synapses into the DMS. And of course, we think that dopamine is playing an important role in updating these learned values. So I mentioned earlier this idea of a select-suppress model for the direct and indirect pathway function. And if I could overlay our task on this model, what we might imagine happens is as mice learn and update the value of odor 1, which is rewarded, we might get increased cortical striatal plasticity for inputs that are relevant to odor 1 that would then be strengthened onto the direct pathway to promote responding to odor 1. Whereas you can imagine as mice get negative feedback and don't receive reward, you might have enhanced plasticity for cues related to all of these unrewarded odors that then may have enhanced plasticity onto the indirect pathway to ensure that mice withhold responding to these unrewarded targets. So we wanted to really test what the function of the direct and indirect pathway was in terms of choice, selection, and suppression. And we wanted to use a tool that would modulate the output of the direct and the indirect pathway, but preserve the strength and patterns of inputs to the striatum that we think might be established through learning in this task. So for that, oh, I forgot to mention, I'm sorry. So with this model, I mentioned that we might expect that you'd have LTP or enhanced plasticity onto both pathways to help promote selection of the high value target. But in vitro slice physiology experiments have shown that following early goal-directed learning, there is LTP onto D1 neurons, which fits this model, but they've observed LTD onto D2 neurons. So that suggests that this model might be oversimplified. So as I mentioned, we wanted to modulate the output of D1 and D2 neurons while preserving the pattern of inputs. So for that, we turned to the chemogenetic tool DREADS that Tiago mentioned yesterday in his talk. So these are modified GPCRs that can be selectively expressed in the direct or indirect pathways. And they come in different varieties that couple to different downstream signaling pathways that lead to either neuronal excitation in the case of HM3DQ or neuronal inhibition in the case of HM4DI when activated by this inert ligand CNO. And for the sake of not losing people in this talk, I'm going to oversimplify and just say excite or inhibit in my graphs. So in the design for this task, we administer a control saline injection prior to training, and then 24 hours later, before we test mice, we administer CNO. And we give it to all of the mice, including the M-Cherry controls, to control for off-target, non-specific effects of CNO. So we had a really basic hypothesis that if D2 neural activity is important for suppressing responding to low-value targets, if we inhibit them during test phase, we might expect to see more unrewarded, incorrect choices. So in light of that model, we were surprised to see that inhibiting D2 neurons in the dorsomedial striatum had no effect on test performance. However, when we excited the D2 neurons, we actually saw increased numbers of incorrect choices and increased trials to complete the task. So this kind of didn't fit with this model that the D2 neurons are really important for suppressing low-value responses. More in line with this select-suppress model, we did see that inhibiting D1 neurons in the DMS impaired test performance. So these preliminary results suggest that this select-suppress model of D1 and D2 function may not be sufficient, or perhaps it's sufficient in explaining the function of D1 neurons, but doesn't explain what the D2 indirect pathway is doing. So we wanted to turn to reinforcement learning to try to gain better insight into what is being affected in the test performance. And we started with standard reinforcement learning models. And at first, we were excited to look at the inverse temperature parameter, given the dorsomedial striatum has been associated with explore-exploit balance in the past. And we saw this increase in unrewarded choices, which might suggest a more exploratory choice policy. We also added this non-standard parameter that I'll just explain quickly. It was a decay parameter. And so what this did was it allowed the ending odor values from training to decay back towards the naive preferences. And this was a step that was implemented once at the beginning of training. At the beginning of testing. So you can imagine in the 24 hours between training and testing, these learned odor values may decay back towards initial preferences. So when we did the standard reinforcement learning models, we had we weren't doing a very good job of capturing test performance, particularly for animals who made unrewarded incorrect choices. And we saw that the performance of the model improved a lot when we added this decay parameter, particularly for the mice who made many mistakes during the test phase. And what I'm plotting here is for each animal, these are all the groups overlaid, the actual trials to criterion in the test phase, and then the mean of all of our simulations we ran based on our model fits. So decay seems to help with this recovery. And we didn't, so far we're still doing our model comparisons, but we haven't found any evidence for learning rate or exploration effects in these groups. So in line with this idea that adding the decay parameter helped with our recovery, we saw that when we looked at the types of errors that mice were making in the test phase, the incorrect choices they made did seem to track with what their naive preferences were going into the task. And we looked and saw that for the D1 inhibitory group, this decay parameter was significantly lower, which suggested that the mice, when we inhibited the D1 neurons, were relying more on their naive preferences rather than the learned odor values. I mentioned that we were initially interested in looking at the inverse temperature, and maybe we were promoting more exploratory choice policy. But looking at the raw behavior data, we have some evidence that the mice are not more exploratory. And so we actually found that exciting D2 neurons or inhibiting D1 neurons significantly reduced the number of entries that mice made during the test phase. And this seemed to be driven by an increased propensity to just choose the first target that they encountered. And I know this isn't exactly the same as an inverse temperature versus physical exploration, but just trying to figure out if those two might be related. So to conclude, we found that inhibiting D1 or exciting D2 neurons leads to increase in unrewarded choices during testing. And inhibiting D1 neurons is consistent with the decay of the learned odor values. And the same mice search less extensively during foraging and are more prone to choose the first target they encounter. So I mentioned this kind of surprising plasticity result that was observed before, where goal-directed learning causes LTP onto D1 and LTD onto D2 neurons. And this didn't really fit with the select suppress model. So what we are thinking is that maybe our data where we might be counteracting this normal learning-induced plasticity and bringing mice back to a more naive state. And sorry for going over. I'd like to thank the Wilbrecht lab, specifically An Collins, who joined UC Berkeley and has been really helpful in doing our model comparisons, and senior undergrad Ben Hoshal, who really helped a lot with the reinforcement learning modeling. Thank you.
Thank you. We have time for one brief question.
Great work. I'd like to push back a little bit on whether or not your data is going against this kind of opposing action channel model. If you're increasing the excitability of the D2 pathways, presumably you're introducing more caution in selecting inappropriate actions. So wouldn't that increase the likelihood of just a random action being selected from the other pathways? So in a sense, it seems like your data is actually in alignment with the opposing pathway model, not necessarily against it.
Yeah, I think what you're getting at is that the data could be consistent with a more careful consideration of the model. We don't have any information about whether we're within or outside of specific action channels. So it would be really amazing to be able to localize action channels and manipulate within or without. Because you can imagine that without would be suppressing just maybe ungoal-directed behavior, just irrelevant behaviors. And then within, you might actually have the opposite effect. Yeah, that's a good point.
Thanks.
Let's send the speaker again.